Everyone, today I will be explaining 20.2 prokaryotes. I will explain how the two groups of prokaryotes differ, and I will also describe how prokaryotes vary in structure and function, and finally explain the role of bacteria in the living world. Microscopic life covers nearly every square centimeter of this earth. The smallest and the most abundant of these microorganisms are prokaryotes. They are unicellular organisms that lack a nucleus. Prokaryotes have DNA, like all other cells, but their DNA is not found in a membrane-bound nuclear envelope, as it is in the eukaryotes. Prokaryotes' DNA is located in the cytoplasm. Prokaryotes are divided into two very distinct groups, bacteria and archaea, two of the three domains of life. The domain bacteria corresponds to the kingdom Eobacteria, and the domain archaea corresponds to the kingdom Archaeobacteria. We will be discussing what is the typical structure of bacteria. The larger of the two domains of prokaryotes is the bacteria. You can find bacteria almost everywhere. They live in fresh water and in salt water, on land, and on within the bodies of humans and other eukaryotes. The structure we have here today is a bacterium called the E. coli. It lives in human intestines, and it has the basic structure typical of most prokaryotes. As we can notice here, the bacteria is surrounded by a cell wall that protects the cell from any injuries, and it also determines the shape. It contains peptidoglycan, which is a polymer of sugar and amino acid that surrounds the cell membrane. It is also noteworthy to note that some bacteria, like the E. coli, has an extra outer membrane that protects the cell from any damage. Prokaryotes also use the flagella for movement and the pili to anchor the bacterium to a surface or to other bacteria. It also has DNA and ribosomes. It is very important to know the differences and similarities between archaea and bacteria. Some traits that they share is that they are equally small, they lack a nuclei, and they both have cell walls. But there are significant differences to mention. For example, the cell walls in archaea lack peptidoglycan. Their membrane contain different lipids, and their DNA sequence of archaea is closer in resemblance to eukaryotes rather to those of bacteria. Scientists also observed that archaea are able to live in extremely harsh environments. As such, some live in environments with little to no oxygen, some live in extremely salty environments or in hot springs, where the temperature would reach boiling points of water. Now, you may be wondering, how do prokaryotes vary in their structure and function? Well, prokaryotes vary in their size and shape, the way they move, and the way they obtain and release energy. Their sizes range from 1 to 5 micrometers. They come in many shapes, like the bacilli, which is a rod-shaped prokaryote, or the spherical prokaryote called cocci. And the spiral and corkscrew-shaped prokaryotes are called spiral. Another thing that can be used to distinguish between prokaryotes is by their movement. Some do not move at all, while others are propelled by the flagella, and some glide slowly along a slime-like material they produce. Prokaryotes need a supply of chemical energy, which they store as fuel molecules such as sugars. The energy is then released from these molecules during cellular respiration, fermentation, or both of these processes at the same time. For example, here we have 4 modes of nutrition. A heterotroph, it takes in organic molecules from the environment or other organisms so they can use it as both energy and carbon supply. And they have a wide range of environments. For the photoheterotroph, they are like the basic heterotrophs, but they also use light energy. And their habitat has to be an environment where there is lots of lights. For the photoautotroph, they use light energy to convert CO2 into carbon compounds, and they have to have an environment where the light is plentiful. For the chemoautotroph, they use energy released by chemical reactions that involves ammonia, hydrogen sulfide, and many others. They have to live in a chemically harsh or in deep environments, like deep in the ocean, in thick mud, in digestive tracts of animals, and in boiling hot springs of water. Prokaryotes also release energy, and there are 3 modes of metabolism that can happen. 1, an obligate aerobe. It requires oxygen, and how it's released is by cellular respiration. It must have a ready supply of O2 to release fuel energy. And their habitat must be oxygen-rich environments, such as near water surface or in animal lungs. Number 2, we have obligate anaerobe. That requires a lack of oxygen, and how their energy is released is by fermentation, so they die in the presence of oxygen. Their habitat must lack O2, such as deep soil, animal intestines, or airtight containers. Number 3, we have the facultative anaerobe, which is surviving without oxygen when necessary. And how their energy is released, they can either use cellular respiration or fermentation as necessary. And their habitat must be oxygen-rich or oxygen-poor environments. For their reproduction and the change of prokaryotes, they reproduce using binary fission. It is when the prokaryotic cell has grown to the extent where it doubled in size, replicates DNA, and divided in half, producing 2 identical cells. If the conditions were favorable in the environment the prokaryotic cell is at, the growth can happen in astonishing rates. But when the growth conditions turn to be unfavorable, prokaryotic cells form an endospore. It is a thick internal wall that encloses the DNA and a portion of the cytoplasm, protecting the cell from harsh environments. Since prokaryotes produce asexually, there are 2 ways a prokaryotic cell can evolve, which is 1, by mutations. They are one of the main ways prokaryotes evolve, and they are inherited by daughter cells that are produced by binary fission. Another is by conjugation. It is a process where prokaryotes exchange genetic information. During conjugation, a hollow bridge forms between the 2 bacterial cells, and the genetic material, usually in the form of plasmid, moves from one cell to the other. Many plasmids carry genes that enable bacteria to survive in new environments or to resist antibiotics that might otherwise prove fatal. This transfer of genetic information increases the genetic diversity in populations of prokaryotes. Prokaryotes are essential in maintaining every aspect of the ecological balance of this living world. They have 3 important roles, which are decomposers, producers, and nitrogen fixers. Prokaryotes assist by breaking down or decomposing dead organisms. They supply raw materials and thus help to maintain equilibrium in the environment. They are also essential in industrial sewage treatment, helping to produce purified water and chemicals that can be used as fertilizers. And these are examples of bacteria that are decomposers. Producers. Food chains everywhere are dependent upon prokaryotes because they are producers of food and biomasses everywhere. And also, nitrogen-fixing bacteria and archaea provide 90% of the atmospheric nitrogen. An overview of our lesson is that the classification of prokaryotes is divided into 2 groups, bacteria and archaea. They are diverse in size, shape, movement, and metabolism. And finally, they are extremely ecologically important. Thank you.